All right. Welcome back, everyone, to the Deep Dive. You know, it's fascinating, right? Today, we're going to be delving into the world of mitochondria.
Oh, those tiny powerhouses.
Yeah, the powerhouses of the cell, right?
Yes.
But you know what's even more fascinating, right?
What's that?
We're going to look specifically at how their, energy-producing core, which is a system called oxidative phosphorylation,
Right.
or, OXPHOS for short, how that changes as we age.
Yeah.
It's like looking really under the hood of our cellular engines as the years go by, right?
It really is. It's fundamental to aging. Like the mitochondrial health, you can think of it like this. It's like the baseline, right, for overall vitality.
Oh, for sure.
For the cell. And OXPHOS is like the main energy producer. So understanding how that changes over time gives us a really crucial window into aging itself.
Absolutely. So today, what we want to do is unpack how these vital mitochondrial components, right, how they're put together,
right,
how they're maintained, throughout life, and what happens to them ultimately as we get older.
Right.
And all of this, of course, is based on some, pretty interesting recent research.
Absolutely.
Okay. So, as we get older, we all know, right, things don't always work as smoothly as they used to. Is that also true for our mitochondria in this OXPHOS system?
Yeah, it's a well-established observation that mitochondrial function, it kind of declines with age.
Right.
And at the heart of that decline is defects in the OXPHOS system. So across different organisms and tissues, we see this pattern emerge. But it's important to appreciate, like, age-related mitochondrial dysfunction, it's not one simple thing. It's like a really complex interplay of many factors we're still trying to fully understand.
Yeah. And of course, mitochondria are involved in so much more than just energy, right?
Exactly.
Not just the ATP.
Right. Yeah.
The cellular oomph.
So while that is critical, ATP production via OXPHOS is critical. They're also metabolic hubs. So they're involved in biosynthesis, building molecules, essential molecules. Oh, wow.
And they're also key communicators within the cell. They influence everything from cell division to when a cell might self-destruct.
Right.
So when we're talking about changes in OXPHOS with age, remember, it has consequences that go way beyond just energy levels.
So when scientists, take a look at aging cells, what are some of the kind of key telltale signs that the OXPHOS system is changing?
So there's several consistent patterns that emerge. Typically, we see a decline in the overall ability of the cell to breathe. So their rate of oxygen consumption goes down. Yeah. And this is indicating, less efficient energy production. The protein machines that drive OXPHOS, the mitochondrial respiratory complexes, these are think of them as like the individual mini factories within the powerhouse.
So their enzyme activity that decreases, the mitochondrial membrane potential, which is that crucial electrical gradient across the inner membrane that's essential for driving ATP production, that also tends to weaken. And not surprisingly, this all adds up to a decrease in the amount of ATP, which is the cell's primary energy currency.
And I remember reading that, along with this energy decline, there's often an increase in those reactive oxygen species, the ROS.
Yeah, that's another really common finding. We often see an uptick in the production of these potentially damaging molecules with age.
Okay.
And then finally, there's often a reduction in the cell's investment in OXPHOS itself.
Yeah.
So lower levels of genes that code for these components. So it's less efficient respiration, sluggish enzyme activity, a weaker membrane battery, less ATP produced, more ROS generated, fewer building blocks for the system.
So really it all comes down to affecting those core OXPHOS complexes.
Absolutely. Yeah.
It's interesting how some of those, I guess, they make sense, right?
Like less activity leads to less energy. But the link between lower OXPHOS and increased ROS feels a bit puzzling to me.
So what's fascinating here is that even if the overall OXPHOS activity is lower, damage to specific parts of the machinery can actually lead to electron leakiness. So imagine, like, tiny sparks flying off, a slightly broken engine.
Oh.
And these sparks are the electrons reacting prematurely with oxygen, generating ROS. So a less efficient system can actually, in a way, be a leakier system.
Oh, that makes a lot of sense. Yeah. So these crucial energy-producing machines, right, these MRCs, they're clearly vulnerable as we age.
Yeah.
But how are they even, assembled and maintained throughout our lives, especially when things start to get a bit worn down?
That's a great question, because the complexity of the OXPHOS system makes it particularly susceptible to age-related disruptions.
Yeah.
These aren't, simple proteins. They're massive assemblies.
with intricate structures. It involves many protein subunits and these essential helper molecules called cofactors.
Right.
So there's many potential points of failure that can accumulate over time.
And it's not just one set of instructions, right?
Yeah.
I remember there's like 2 different sources of genetic information involved.
Exactly. The creation of these OXPHOS complexes is unique because it requires a highly coordinated effort between the cell's main control center, which is the nucleus with its nuclear DNA, and the mitochondria themselves, which have their own small circular genome, mitochondrial DNA, or mtDNA.
So the nucleus provides the majority of the blueprints, but the mitochondria have their own specific set.
Precisely. So the mitochondria have their own machinery for copying their DNA, transcribing it into RNA messages, and then translating those messages into specific OXPHOS subunits.
Wow.
They even have their own version of ribosomes, called mitorobosomes, which are distinct from the protein-making machinery in the rest of the cell.
So as cells age, this balancing act, this delicate balancing act between the nuclear and mitochondrial systems.
Exactly.
To make sure all the right parts are produced at the right time in the right amounts, that could become a major challenge.
Yeah, that's a key area of ongoing research. There's evidence that suggests that cytoplasmic translation, which is the process of making proteins from the nucleus-derived RNA in the main part of the cell, that might become less efficient with age. We see indications of problems with ribosomes themselves and this disconnect between the levels of RNA instructions and the actual amounts of protein being produced. So keeping everything in sync becomes increasingly difficult.
And what happens to those protein pieces that are made, but they don't immediately get incorporated into a working complex?
Yeah.
Are they just floating around?
No. There's quality control mechanisms. So those unincorporated or misfolded subunits are tagged for degradation. So ensuring that faulty components don't gum up the works. But interestingly, under normal conditions, some OXPHOS components are actually produced in surplus.
Really?
Yeah. So there's a built-in reserve to ensure proper assembly, even if some parts are damaged or lost.
So the mitochondria have a way to handle some level of imbalance. But as we age and more things potentially go wrong, is that cleanup capacity enough? Can it get overwhelmed?
Right. That's the big question. So when the OXPHOS system is disrupted, cells can activate these stress response pathways. Things like the mitochondrial integrated stress response, the ISRMT, and the unfolded protein response, the UPRMT. These are the cell's attempts to restore balance and repair damage. However, the effects in aging are complex. They can be contradictory. It depends on the organism. It depends on the stress. We see hints in simpler organisms that mild mitochondrial stress and activation of UPRMT can actually extend the lifespan.
Wow.
But in more complex organisms like mammals, significant OXPHOS defects, they lead to disease.
That leads us to the topic of mtDNA mutations. I know those are often talked about. In the context of aging mitochondria.
Absolutely. So the accumulation of what are called somatic mtDNA mutations, meaning genetic changes that happen during your lifetime. Not inherited from your parents. This is a very common observation in older individuals. And these mutations, they can range from, single letter changes in the DNA to larger deletions in the mitochondrial genome.
And what's the primary source of these mutations?
Well, the prevailing theory is that they arise from errors when mtDNA is copied. So a process called replication. And this idea is strongly supported by studies in mice, especially bred mice, that have a much higher rate of mtDNA mutations. So these mtDNA mutator mice, they exhibit signs of accelerated aging. So there's this direct link between these replication errors and aging. And these mutations, even though they're in a small genome, they can have a huge impact on energy production. They can lead to faulty OXPHOS subunits.
So now I've encountered the term heteroplasmy in this area. What exactly does that mean?
Heteroplasmy describes a situation where you have a mix of mtDNA molecules. So within a single cell or even a single mitochondria, and you don't just have one uniform version. So some copies might carry a specific mutation while others are normal. And the level of heteroplasmy refers to the proportion of mutated mtDNA in that pool. And the effects of a mutation often don't become apparent until it reaches a certain threshold. And also because mtDNA replicates kind of independently of the cell cycle. A mutated copy can be preferentially replicated in some tissues, leading to an increase in the proportion of mutated mtDNA over time. This is called clonal expansion. The number of mtDNA copies in a cell can also be a factor. So it's a dynamic picture influenced by the tissue, the environment, and the individual's nuclear DNA. But the accumulation of these mtDNA replication errors is a central concept in understanding the age-related decline of OXPHOS.
Okay. So we've got the blueprints and the potential for errors.
Right,
in those blueprints.
Yeah.
But even with good blueprints, even with perfectly good blueprints coming from the nucleus, the protein components still need to be transported into the mitochondria, right? That's the import process.
The import of the vast majority of mitochondrial proteins, including many OXPHOS subunits, this is a crucial step. And any problems, any hiccups in this process can trigger cellular stress responses.
Oh, yeah.
There's significant crosstalk between the protein import machinery and the OXPHOS system itself. So if OXPHOS isn't running efficiently, it can actually impede the import of new components.
So it can become a vicious cycle where poor energy production is hindering the delivery of the machinery needed to fix it.
Precisely. So these nuclear encoded OXPHOS components are initially made in the main part of the cell, the cytoplasm, as unfolded precursor proteins. They then have to navigate their way into the mitochondria, passing through these protein channels in the outer and inner mitochondrial membranes. So we're talking about the TOM and the TIM complexes. And this journey is often guided by a specific signal sequence at the beginning of the protein. The actual movement across the inner membrane requires energy in the form of, the mitochondrial membrane potential and ATP hydrolysis. In the space between the two mitochondrial membranes, the MIA pathway plays a role in helping some imported proteins fold correctly. So this process involves oxidation and reduction reactions. It's energy intensive and a finely tuned process that relies on a healthy mitochondrial environment. Stable membrane potential, availability of specific molecules.
So if this import process isn't working correctly, what are the consequences?
Well, mutations in the genes that encode the components of this machinery have been linked to premature aging syndromes.
Oh, wow.
In humans. So it's really highlighting how important this process is. We know that in normally aging cells, there's often a reduction in mitochondrial respiration and membrane potential, which could impair the import of new OXPHOS components. But mitochondria have a clever backup system. They can actually run the ATP synthase enzyme in reverse.
Wow.
Using ATP to pump protons and maintain that crucial membrane potential. But this comes at a cost, right? Energetically, it's costly. But it suggests that cells prioritize maintaining this potential because it's so essential for other mitochondrial functions, like protein import.
So once these precursor proteins successfully get inside the mitochondria, they're not quite ready to go, are they? There's still some finishing and assembly required.
Once they're inside the matrix, these precursor proteins have their signal sequences snipped off by enzymes called peptidases, like MPP. Then they need to fold into their correct 3D shape. And this is often assisted by chaperone proteins, such as MTHSP70 and LONP1. If LONP1, for example, isn't working properly, newly imported proteins can aggregate in the matrix. While protein aggregation is a hallmark of aging and some diseases like Alzheimer's, we don't have a complete picture of whether the OXPHOS components themselves are prone to aggregation and aged mitochondria. But the presence of chaperones, it suggests a significant capacity to prevent this misfolding.
And then comes the actual putting together, these building blocks into those functional respiratory complexes.
The assembly of MRCs is a highly coordinated step-by-step process. It doesn't just happen spontaneously. It's orchestrated by a large team of dedicated assembly factors. So these are proteins that aren't part of the final complex, but they're essential for its formation. They help bring together the different protein subunits, incorporate the cofactors, and ensure everything is in the right place before you have the complete functional MRC. And this assembly process is also tied to the synthesis of those mtDNA-encoded subunits. And is overseen by a quality control system that includes these protein-degrading enzymes within the inner mitochondrial membrane. So when this assembly line malfunctions, whether it's due to faulty faulty subunits or problems with the assembly machinery itself, it's implicated in diseases, Parkinson's, Alzheimer's.
So aging can disrupt this complex assembly process at multiple points.
Precisely. So the accumulation of mtDNA point mutations can directly affect the structure and stability of the mitochondrially made subunits, which can then interfere with proper complex formation.
Right.
And the availability of essential cofactors like iron-sulfur clusters and the stability of the complexes can be influenced by the cellular environment. Things like oxygen levels and age-related modifications to proteins. We've even seen examples where stress, like aging, can trigger a change in the composition of these complexes.
Oh, wow.
Where different versions of certain subunits might be incorporated as a way to adapt.
So the cell is trying to swap out parts to keep the engine running as best as it can.
Right, exactly.
Then we have these higher order structures. We have these super complexes.
Yeah.
What's their role in all this?
So the MRCs can further organize themselves into larger assemblies called supple complexes.
Okay.
With the respirescent being the most well studied. So this is a combination of complex 1, 3, and 4.
Okay.
While we know these things exist, their precise function and the advantages they offer are still under investigation. One idea is that they might facilitate a more efficient transfer of molecules between the individual complexes.
Okay.
Like an assembly line, right?
Okay.
Where the parts move smoothly from one station to the next.
Yeah.
They might play a role in regulating the assembly of individual complexes, influence ROS production, and affect metabolism. And the composition can change depending on the metabolic needs of the cell.
Wow.
But while it might seem like these larger structures are essential, studies have shown that the MRCs can function pretty well even without being tightly organized into supercomplexes.
So why bother forming them at all if they're not strictly necessary?
Right. One compelling hypothesis is that it might provide stability to the inner mitochondrial membrane, so preventing the individual complexes from unfolding or aggregating, which could be important as the cellular environment changes with age.
Right.
But the evidence about supercomplexes and aging is still somewhat limited.
Okay.
It's largely correlative.
Okay.
Making it difficult to definitively say what their role is in aging.
Got it.
We also see other higher order structures.
Okay.
Like pairs of complex V enzymes.
Okay.
Which are important for shaping the membrane.
Right.
And those structures can be altered in aged tissues.
So it sounds like the mitochondria have a lot of quality control built in.
Yeah.
What about the actual breakdown and replacement, the turnover of these respiratory complexes?
Right. So given that impaired protein turnover is a characteristic of aging, right, across the cell, mitochondria have their own dedicated quality control systems to deal with damaged components. So one key aspect is the dynamic nature of the mitochondrial network itself.
Okay.
The fact that they can fuse together and fragment, this helps to mix the healthy and damaged parts.
Right.
Or isolate dysfunctional mitochondria.
Okay.
These damaged mitochondria can be targeted for removal through things like mitophagy, which is a specialized form of autophagy that targets mitochondria, as well as other degradation mechanisms.
Nice.
So while defects in mitophagy have been linked to early onset Parkinson's disease, the role of some key mitophagy players in maintaining healthy OXPHOS in normally aging organs is still an area of active research.
So it's not just getting rid of entire damaged mitochondria, right?
Right.
What about replacing individual worn-out parts?
Right. So this is where we get to the concept of heterogeneous turnover of the mitochondrial proteome.
Okay.
Different mitochondrial proteins have different lifespans, even within the same OXPHOS complex. Some subunits can be remarkably long-lived in certain tissues, suggesting precise mechanisms to manage the turnover of other components.
Oh, wow.
And interestingly, the turnover rate of some OXPHOS components has actually been observed to increase in aging brains.
Really?
Yeah. This asynchronous turnover suggests that proteases within the mitochondria play a role in removing damaged OXPHOS subunits. So it's a targeted maintenance strategy rather than just wholesale degradation.
Let's circle back to ROS for a moment. We know it increases with age, but it's not always the enemy.
Right. That's an important point. So the old idea that aging is simply a result of accumulated damage from mitochondrial ROS, that's largely been overturned. We now recognize that ROS can be signaling molecules.
Okay.
And can be beneficial depending on the type of ROS where it's produced and the cellular environment.
So where exactly does this ROS come from within the OXPHOS system?
So the primary sites are complex I on the matrix side and complex STIR on the intermembrane space side. It's often assumed that damaged MRCs or reduced levels of these complexes are the main drivers of increased ROS, but the relationship is really complex and sometimes counterintuitive. Elevated ROS can also be part of normal metabolic adjustments, adaptations to stress, and even signaling pathways.
And I've heard about something called reverse electron transport being involved in ROS production.
Yeah. So reverse electron transport, or RET, through complex I, this is a process where electrons are forced back along the chain, which can boost ROS production.
Wow.
This mechanism has been implicated in various conditions, like when blood flow is restored to tissues after oxygen deprivation, so ischemia, reperfusion injury, and in inflammatory responses. And fluctuations in tissue oxygen levels, which are common in age-related diseases, can also influence OXPHOS and ROS generation via RET. So, for instance, a period of low oxygen followed by a sudden return to normal oxygen levels can trigger this. And interestingly, some compounds with potential anti-aging effects, like metformin, might actually work by counteracting this reverse electron flow and the associated ROS production. There's also evidence of alternative electron transfer pathways that become more active under low oxygen conditions, where molecules like fumarate can act as alternative electron acceptors.
Finally, let's consider how these changes in OXPHOS might differ across various parts of the body and how they relate to the development of age-related diseases.
Right. So OXPHOS function isn't one size fits all across the body. There's variation between cell types, organs, and even within a single cell.
Yeah.
For example, during blood cell development, stem cells that reside in low oxygen niches give rise to more metabolically active progenitor cells through a process that involves the segregation of older mitochondria.
Oh, wow.
We also see that age-related OXPHOS alterations are not uniform.
Right.
So tissues that have a high energy demand and don't divide readily after maturity, like the heart and brain, these are particularly vulnerable. Many age-related diseases like heart failure, muscle loss, neurodegenerative conditions like Parkinson's and Alzheimer's, and kidney dysfunction have been linked to impairments in OXPHOS.
Whoa.
And even within an organ like the brain, the way OXPHOS is affected can differ between different cell types.
Oh, wow.
Contributing to the disease in distinct ways. There's even a concept of hypermetabolism in aging, where less efficient OXPHOS leads to increased energy expenditure through these compensatory stress responses, potentially contributing to the overall aging process.
It's truly remarkable how intricate all these processes are. So, as we begin to wrap up this, deep dive into the fate of our mitochondrial powerhouses, what are the main things, we should take away?
Well, it's clear that changes in the OXPHOS system are a fundamental aspect of aging.
Yes.
And our understanding of these changes is, constantly being refined. We're learning more about how these energy producers are assembled, maintained, and transformed.
And there are still, it sounds like, many pieces of the puzzle that we're working to fit together.
Absolutely. So many questions about the exact sequence of events and the roles of different molecular players, especially in the complexity of the human body.
Yeah.
But the future of research in this area is promising. We have sophisticated new technologies that'll allow us to probe these mysteries at an unprecedented level.
So as we age, these incredible powerhouses within our cells, they undergo this complex and fascinating journey of changes.
Yeah.
Influencing everything from our daily energy levels to our susceptibility to these age-related conditions. It makes you think, how this interplay between our genes, our environment, and all these intricate processes really shapes the health and longevity of these vital energy producers within us. And what are the implications of understanding?
Yeah.
Potentially influencing these mechanisms as we continue to age.
It underscores the complexity of life at the cellular level and this quest to decipher these processes that govern aging.
Absolutely. So this has been a really insightful deep dive. Thank you so much for sharing your expertise, guiding us through this complex landscape.
It's been my pleasure.
So please don't forget to like the video and subscribe if you want more content like this.